let's say that we do a total acid hydrolysis on the tripeptide and we get glycine, alanine, and aspartic acid. Now, if you've been starting to do the problems, you know what he does is he gives you a bunch of blanks, and you have to start filling in the blanks. Or even if you can't fill in the blanks, you should at least say, you can only fill in the blanks when you know the order. So here we don't really know the order, but we could at least start saying what were the amino acids that made up the tripeptide. So what do we know? What do we know about who were the amino acids that made up the original tripeptide?
We definitely have a gly and an ala.
Although we don't know what position they're in.
Right.
And it could have been as or acid.
Excellent, that's good that you remember that. That's right. So the correct answer here would be that the original amino acids were definitely glycine, definitely alanine, and either aspartic acid or asparagine.
That's because there's absolutely no indication of NH4+, whether it's present or not, right?
Yeah, well, you could probably tell which one it was if there was NH4+.
If there was NH4+, then you'd know it was ASN.
That's true.
But he doesn't always, what he does a
Yeah.
lot is he has ASN and GLN. So the NH4 could be coming from either one of
good.
those.
That's better. All right, so let's say we had,
Opposed to not saying anything and then we don't know if he means like that it's
Right.
not there or that he just didn't mention it, right?
Yeah.
It gets confusing.
so this would be a better example. Let's say we got these three.
Gly, as, and glue.
Yeah, what could the original amino acids have been?
So, glue or glyn.
Right?
Aspartic acid.
And glycine. All right, so now we're going to see how we can use hydrazine to tell which of these we started with. Well, to save time, I'm not going to draw the whole tripeptide. I'll just draw one of the amino acids.
So wouldn't the whole point, so wouldn't he want to put NH4+ there for us to start asking the questions of like, you know?
Yeah, I'm still not doing a very good job. I should have said,
No, I was just wondering. because
I should have said that after the
right?
total acid hydrolysis, we got these
Okay,
products.
cool.
I should have said that. That's much better. That's a very important point. I should have said we got these products. And that tells us that one of these probably was not the acid, one of these probably was an amide on the side chain. That's
If he
right.
doesn't say anything, can we be safe to say that he means there is no NH4+ if he just doesn't write?
I believe that's the case, although the best way to tell is by doing all the problems and seeing if he sticks to that. But I believe that would be the case.
Yeah, I mean, I think from some of the older exams, he doesn't do a lot of stuff, which is the problem of doing those old things.
Oh.
Yeah.
In the first couple of finals, he doesn't even have any of these types of problems. So
No, he has incredibly hard ones.
not on the first two finals. So, or maybe your book. Well, yeah, on the first two finals, he didn't even have the peptide analysis like
no,
this.
but
So.
I wanted to ask you how in God's name we're supposed to do one of the first ones.
Okay, well, we can go through those in a second. In any case, let's say that we start with this. Now, this is aspartic acid. And maybe we should also think about this. Which is asparagine. Now, let's say that we use hydrazine. If we use hydrazine, will hydrazine attack here? No. Or here? No. Or here, but will it attack here? Yes. So which of these will have a hydrazine on its side chain? Only asparagine. So if you see that after the total acid hydrolysis, you've got a hydrazine on the side chain, you know that came from originally attacking asparagine and not aspartic acid. That's how we can use the hydrazine to figure out.
If you use hydrazine.
That's
not
right.
Total acid hydrolysis.
That's right.
Okay.
We're using, now notice that the hydrazine is kind of an alternative to total acid hydrolysis the whole point of total acid hydrolysis is to break apart all the amide bonds, but hydrazine also breaks apart all the amide bonds. So usually if you use hydrazine, you don't need to do the total acid hydrolysis at the same time, so you've already split apart all the amide
So in
bonds.
terms of doing like an experiment, you could do, the first part would be doing total acid hydrolysis, and it's like asking, what's the problem?
Mhm.
It's like, well, we don't know.
Right.
And then he would use hydrazine, right? And we'd be like,
Starting with the
Oh.
fresh, yeah, starting with the connected peptide again, that's
got
right.
it, okay.
That's right.
Is each experiment a new kind of, okay, when we.
You have to read
questions-
carefully to see what he's treating.
Yeah,
Yeah, we need to do problem.
yeah. so
Okay, but anyway, we're seeing here how hydrazine works. Normally, if you would see these results, you don't know which of these came from the amide side chain. But if you see a hydrazine on the side chain, you know that came from an amide side chain. It didn't come from an acid side chain. That's the basic idea of how we can use hydrazine to distinguish between these.
So it was acid, and then therefore it has to be, so it's acid, that means that one used the NH4 plus, right?
We also have to look at what happened at the
So I'm assuming that these were both at the, to make things simple, I'm assuming these were both at the C-terminus. So let's say these amino acids were at the C-terminus, so we have a carboxy group over here. Well, if you treat aspartic acid with hydrazine, you would get this. But if you treat asparagine with hydrazine, you would get this. So now we can work backwards. If we use hydrazine and we see this product, we know it came from asparagine. But if we use hydrazine and we see this product with a carboxylate on the side chain, then we know that came from aspartic acid. So hydrazine gives you two different products. It gives you one type of product when it attacks aspartic acid, and gives you a different type of product when it attacks asparagine. So we can tell which of those we started with. Does that make sense?
That makes sense. And then what does it tell us for, then can we assume that the NH4 plus was either yes or not used for glue instead of doing any other experiment with glue?
Well, for example, let's say that what we actually got was this.
Yeah.
If we got this, then we know that the original amino acid was aspartic acid, because this comes from aspartic acid.
Right.
So I can cross out this possibility. We know that originally we started with the aspartic acid here.
Okay.
And therefore, where could this ammonium have come from? Well, now the ammonium must have come from glutamine.
So if he gave us 2 equivalent of ammonium, then we would.
Then things would be harder.
Yeah, I've never seen them do that.
Yeah, I'm just.
Okay, if you want, maybe we should look at an actual problem now to see how
Yeah, I
these
think
work
we should do that. That'd be great.
in action. Why don't you pick a problem to look at?